neurotoxicity. His pioneering He has contributed significantly to molecular neurobiology and toxicology. And I would like to invite our respected chairperson, Dr. Amir Nazir, and grace the dais. Dr. Amir Nazir, Senior Principal Scientist at CSIR CDRI Lucknow, Head of Toxicology. He leads IND enabling regulatory studies and GLP program. Now I request two session chairs to start the session.
शुभम देखते हैं
Very good afternoon to all of you. Before we start the talks, on behalf of my co-chair, Dr. Amir Nazir, I take this opportunity to congratulate the organizer, especially Dr. Shalini Tripathi, and the whole team for taking this initiative to organize this conference and to schedule all the presentations. Because I had attended all the presentations in the morning. It was a good learning for all of us. I request you all to join me in giving a huge round of applause to Dr. Shalini and all her team in all sincerity. Because we are running behind as far as the schedule is concerned, so I will not go into the flowery talk, lovely words, but will come to the main point and will request, because earlier one of the speakers was not there in the earlier session, but here I am sure that all the speakers are there. I will request them, if they are able to and they are able to accept my request to reduce the timing of their presentation, because they all are very seasoned players, I am sure. If they are able to reduce their time of the talk to 5 to 10 minutes, so that we will be able to cover. So in this session, I have a lot of pleasure to invite the first speaker. He is Professor Rajiv Jain. Dr. Rajiv Jain has been associated with the Jivaji University Gwalior, and he was the project coordinator and the principal investigator of the DST at New Delhi, and he was former professor and head of the Department of Chemistry at the Jivaji University. And Professor Jain also has been associated with Central University Pondicherry as Pro-Vice Chancellor, and he has over 375 publications and has an H index of 60 and has supervised over 100 PhD scholars, and he ranks among the top 2% scientists of the world as far as the Stanford University scale is concerned. He is currently serving as project coordinator of a DST outreach initiative to promote scientific temper through popular science education and chemistry demonstrations. I will not come in between you and Dr. Jain, and with these words, I request him just to deliver the talk. Sir, we will appreciate if you are able to cover the talk in 20 to 25 minutes.
Definitely. Good evening. It is my pleasure to be with you, to interact with you under the chairmanship of Professor Khanna and Professor Nazir. So I shall speak on a somewhat different topic which you have, which we are discussing from the morning. It is a topic related to drug development, but it has very important role in the development of drugs. The techniques which you use for the identification, for elucidation of structure, for identification of intermediates are generally spectroscopic technique and chromatographic technique. That technique you use for the detection of different types of products which you form. But and also by incorporation of different types of nanomaterials, particularly graphenes, carbon nanotubes, carbon nanomaterials, and fullerenes, different types of, I am showing here, these are single bond carbon nanotubes, multi-bond carbon nanotubes, graphenes, different forms of carbon nanomaterials we are using. I will show a number of slides here how we are using them for the sensitive determination of products formed at every stage of clinical trial, starting from the screening to the final product development. And during that process, during impurity profiling, we could identify new products which are more important than the products which we were trying to synthesize. Similarly, by electro-organic synthesis, we could synthesize molecules which were not possible to synthesize by classical methods. These are supplementary techniques, or we can say even by exclusive use of electrochemical technique, we can identify substances. Simultaneously, we can quantify them, we can find out their concentration, and also we can identify the intermediates by electro-organic synthesis by using different types of electrodes, and we can use these electrodes in the form of sensor, for sensors for navigation and synthesis of different types. In contrast, chemical sensors offers a unique combination of sensitivity, selectivity, miniaturization, and real-time monitoring, making them indispensable in modern pharmaceutical research. A chemical sensor, if I define it in a simple word, is a device that converts chemical information such as concentration or presence of a specific analyte into an analytically useful signal. For the identification of some substances which you have synthesized, or for the purification purposes, generally you use chromatographic method. You use HPLC or LCM. Or for identification you use spectroscopic technique, NMR and mass spectrometry. But here we are supplementing all these techniques, or exclusively we are using electroanalytic techniques, which is a group of 60 different techniques based on a very simple principle, very simple techniques, and require no sample preparation, almost no sample preparation. For any chromatographic method, for quantification, you require a exhaustive sample preparation technique. For spectroscopy, your sample should be very clean, with no interference. But here, in our technique, electroanalytical technique, no sample preparation is required. Even in the presence of different analytes, you can analyze your analyte, you can monitor the progress of your analyte in the blood or urine or in any sample, and also quantifying them. I will show an experiment which I have done in my lab, that by inserting a miniature electrode in the brain of a rat, we could identify different reactions of the nucleus. So many things are present there. As a biologist, I do not know much, but so many things are present there. Even in their presence, we could identify different types of neurotransmitters. With this technique, which I am presenting here, are very useful for in drug development. These are different types of sensors are there, electrochemical sensor, which is based upon the conversion of applied energy into measurable form, such as current. Then mass sensitive sensor, thermal sensor, chemical sensor, and biosensor. Then why we modify the surface of sensors? We modify the surface of sensor because transfer of physical chemical properties of the modifier to the electrode is increased. Enhanced electrocatalytic activity due to the use of materials with large surface area, which in turn allow better sensitivity. Selectivity towards analyte due to immobilized functional groups and dopant. Fast diffusion kinetics in case of some material extraction and accumulation of an analyte at the surface of the electrode. We can functionalize these nanomaterials which I have shown here. We can introduce different types of functional groups there, which will make them more sensitive. Up to nanograms, up to picograms, there is no problem in identifying the substances and monitoring them. Not only identify, in one experiment we can identify the substance as well as we can find out its concentration. This is the rare one. Usually you do. And also we can identify and quantify 3 or 4 or 5 substances simultaneously. Whereas in your spectroscopy technique, you identify only one substance, whereas in chromatographic technique, you can identify 2 or 3 substances, but sample preparation is required. This is simultaneous determination which I discussed with you. This is the drug development. Generally, following steps are involved. You know very well, all of you know, even that I have written here. This screening, preclinical testing, formulation and quality control, clinical trials and therapeutic monitoring. Chemical sensors play an important role at each stage of drug development, improving their sensitivity and selectivity. So these techniques, if you read a simple Faraday laws of electrolysis, these are based upon Faraday laws of electrolysis, that you apply potential, you apply energy, because in all these techniques, starting from melting point and boiling point determination, NMR, IR, FTIR, any technique, the only one thing is common, that you are applying energy. What may be the form of energy? It may be electromagnetic radiation, it may be heat in boiling point and melting point, it may be thermal analysis, it may be different types of things, radioactive, different types of things are there, but you are applying energy. And after applying energy, you are measuring current or absorbance or any other quantifying data, means sensor is converting your output into measurable device. So during preclinical testing, sensor help in quantifying developing drugs in biological fluids, studying stability, solubility, and degradation pathways of the drug, monitoring synthesis, intermediates of intermediates or impurities. Miniaturized sensors allow the real-time analysis without extensive chromatographic methods. So in every stage of drug development, we are using these electroanalytical techniques for what purpose? For impurity profiling and also for end product analysis. And if some product is like that in your synthesis, which cannot be synthesized by classical method or difficult to synthesize by classical method, in that case we may apply electro-organic synthesis and the electrode. These are electrochemical sensors, voltammetric sensors, amperometric sensors, and potentiometric sensors. Nanomaterials for sensor enhancement, which as I have already discussed. These functionalized nanocomposites enhance selectivity for specific drugs such as antibiotics, anti-cancer agents, and neurotransmitters. These are some data which is not clear here. On the one side, I have taken this is the so on the first column, it is the functionalized sensor. Means it is the working electrode of carbon nanomaterial which has been functionalized. And in the second and third column, its sensitivity has been shown. Examples of chemical sensors in drug development. These are the different types of voltammetric sensors, CNT-based biosensor, and molecularly imprinted polymer sensor for enantiomer selective detection of disease. Another important thing is development of different types of enantiomers. That here, this technique can also identify in confirming the enantiomer and also different conformers. This is the one of the examples of this I have done in my lab, which I have tried to present here. The preparation of a functionalized or modified fabricated multi-watt carbon nanotube for voltammetric sensing of 2,3 silated glycans. Glycans are markers for the detection of cancer, which have been determined by this technique. Here a working electrode is taken, and that one, what is the working electrode? Working electrode is a classic carbon electrode, means sensor, and that a nanosensor has been casted, and that sensor has been functionalized, and that reacts with a target, and that target is alpha-2,3 silated glycine, which forms a complex, and that complex identifies the alpha-2,3 silated glycine, which has been shown here in the form of a complex form, and a cyclic voltammogram is recorded. Then this is the another example of functionalization. How functionalization is achieved? Functionalization of carbon nanotubes, graphene nanoribbons. Here nanoribbons has been taken, which are being functionalized, and it has been used for the voltammetric determination of 2-aminophenol and 4-aminophenol. This is cyclovoltammogram has been shown, which is clearly shows two peaks. One is for 2-aminophenol and another is 4-aminophenol. This is the another example, schematic preparation of scheme of different types of another product, which is used for the identification of its high parameter. So these all experiments I am showing here. I will skip out them. These are the examples how this electrodes, how these sensors can be modified, or these sensors can be functionalized. And their preparation and functionalization is not difficult. It is very easy. It is very, very simple. It is very simple in comparison to the sample preparation. In chromatography and spectroscopy, first stage, the primary stage is the sample preparation. Whereas the time which you will consume in sample preparation, in that time this whole experiment can be done. And you can find out the concentration of unknown substance, and here the mechanism is proposed. And how I have proposed the mechanism, how mechanism can be proposed? It is on the basis of cyclic voltammetry. That we can do cyclic voltammetry depending on the scan rate. We can identify the intermediates having a life of fraction of second. That intermediate can be identified. Whereas in cryoscopic technique, it is difficult to identify that intermediate or not possible. It is only possible in cyclic voltammetry using different types of sensors. Biosensors in drug development. Applications include measuring enzyme inhibition in toxicity studies, monitoring metabolites and biomarkers in clinical trials, detecting therapeutic drug damage in patients, example glucose, antibiotics, antipaprika. The emergence of DNA-based sensors enables the study of drug, DNA interactions, and genotoxicity evaluation. This is another example of determination of biosensor by taking a different using biosensor, lipid cytosine, and detection of another. This is functionalization. Functionalization of single bond carbon nanotube for voltammetric determination of myosin, myosin and butane. This example I have taken for the determination of direct probing the material present in the brain of a rat. I can skip on this. So another type of sensors are optical sensors. Optical sensors are used where in optical sensors absorbance, fluorescence or chemiluminescence principle is used to detect drugs or biomarkers. They are particularly useful for real-time monitoring in microfluidic systems, level-free assays based on surface plasma resonance, in vivo imaging for drug biodistribution. Then integration with microfluidics. The advantages include minimal reagent consumption, fast response time, parallel analysis of multiple samples. This is the beauty of the technique, that multiple analysis, multiple qualitative as well as quantitative analysis, intermediates detection, whose lifetime is fraction of second, and electroorganic testing. All can be achieved in a very small time. These systems combine electrochemical detection with microfluidic control, and the result is very important. Then recent development. I have shortened the lecture. Professor Dr. asked for 20 minutes. I think I have completed in 10 minutes. In 30 minutes, then I have to do recent development. Then wearable sensors are coming. And this is the beauty of the wearable sensor. If we could have wearable sensor at the time of Bhopal gas disaster, then we could have identified what was the intermediate formed then and there. But now it is possible. Now we design wearable sensors for continuous drug monitoring, insulin and antibiotics. Sometimes these are required for monitoring and insulin 24 hours continuously or more hours, and also for monitoring different types of biomarkers found in the body during the cardiac problem. All that can be done. The implantable sensors for pharmacokinetic studies, artificial intelligence, integration for pattern recognition and data analysis. Then green sensor development using biodegradable or reusable materials. What are the future challenges? That is the biofouling and stability of biological components, standardization and regulatory acceptances, the scalability for mass production. Future outlook: chemical sensors may transfer pharmacological research by enabling real-time, sensitive and cost-effective drug analysis. The integration with nanotechnology, microfluidics and artificial intelligence will further revolutionize the personalized medicine and precise physiotherapy. Pharmacotherapy. This is it.
Thank you, Professor
If there
Jain.
is any question, I shall be very happy to answer.
Thank you, Professor Jain, for accepting the request and keeping the presentation on time, well on time. If there is, like as a customary, we don't take up the questions for the plenary talks, but if there is any question, one or two, is from Dr. Jain, we will be very happy to respond to the question. Any question from the back? From other colleagues? If there is not any question, so we will like to thank Dr. Jain. And once again, please join me to give a huge round of applause. Now I will request my co-chair Dr. Ahmed just to introduce the next.
Thank you, Dr. Khanna. It's my pleasure to now introduce the next speaker of the session, Professor Vishal Trivedi. He is a leading structural biologist and biochemist, currently serving as a professor at IIT Guwahati, where he heads advanced research in malaria drug discovery and host pathogen interactions. He earned his PhD from CSIR CDRI and completed postdoctoral training at Harvard University and Tufts University, USA. His work focuses on identifying novel therapeutic targets through integrated protein biochemistry and immunopathology. May I request Professor Trivedi to please deliver his talk. Again, the same request, Professor Trivedi, if you could keep it between 20 and 25 minutes. Yeah, thank you.
Thank you, Amit, and thank you, the organizers, for giving me an opportunity to discuss my research. So today we are going to discuss about the target-based analysis of artemisinin action in host and the plasmodium falciparum, right? And with this talk, the emphasis would be more to understand when you give a drug to a patient or to a person. It does not even go and kill the parasite. It also do many other things. And if we understand the both components, probably we would be able to develop the better drugs. So starting with the malaria, right? So malaria is a malaria causes severe damage to the human society. And if you go by the malaria facts, right? Malaria is transmittable disease which is caused by the mosquito, and it actually spreads when the mosquito bites from the infected person to the non-infected persons. And approximately 97 countries and the territories are being infected with malaria, which are widely distributed throughout the world. And as per this work, like 3.2 billion people are at the risk of getting the malaria. And the number is so big that you can imagine that every 60 seconds there is a kid who is going to get infected with malaria in Africa. So it is a big problem, not only for the for our human society, and the more risk factor is for the small kids and the pregnant women. And because of these facts and because of this such a deadly disease, there is a quote from the Bill Gates which says that the malaria parasite has been killing children and sapping the strength of whole population for tens and thousands of years, and it is impossible to calculate the damage which is being caused by the malaria. So malaria is being caused by malaria in humans are being caused by the multiple species such as malaria, Plasmodium falciparum, Malarium ovale, Knowlesi, and all that, right? But when you see the number of cases, right? If you see the number of cases, and if you see how many cases are Plasmodium falciparum specific versus the other strains, what you found is that over the course of time, since we developed the different types of drugs, the cases of the malaria are reducing over the course of time, but the cases with the Plasmodium falciparum is increasing. Or I will say, the mortality of the person or the mortality of the people is increasing with the Plasmodium falciparum, but the overall burden is reducing. So basically, it is the Plasmodium falciparum which is the main culprit, which is responsible for the death associated with the malaria. And that is because the malaria is having a very complex life cycle. It is a digenetic parasite, so it has one component which is in the invertebrate host, the other component which is in the vertebrate host. And I am sure everybody sitting in this room probably could have studied the malaria life cycle, and that itself will tell you that how important this disease is, because when you were in high school or when you were in 11th standard, at least I remember when I was an 11th standard kid, the UP board's textbooks were having the malaria life cycle. Okay, and when you go to the protozoa, it has a life cycle of malaria and life cycle of the anemia, right? So that is because this disease is so important, and they want to teach and they want to make the awareness about this particular disease. So talking about the different stages, right? The blood stages, where the parasite is forming the different types of stages, starting from the ring to tropho and Schijon, and then Schijon bursts and releases the merozoites to start another cycle of Arctoceratosalpingoni. This is the stage which is very, very important and very, very responsible for the death what is been associated with the malaria parasite. Now, looking at the proteome, right, of these stages, what you see is that the parasite is expressing the different types of proteins which are stage specific and which are actually being diversified starting from the sporocyte to the to the trophocyte and ring stages and all that. But what you see here is that there is a very large chunk of proteins which are actually being categorized as the hypothetical proteins. Now, these hypothetical proteins actually attract the scientists. They think that these hypothetical proteins probably could have some role which probably we don't know, right? And there could be some drug targets which actually can be fetched out from these hypothetical proteins. Now, talking about the drug development, and traditionally the malaria parasite, people were developing the drugs starting from a very old quinine to the latest molecule, which is artemisinin. Now, what you see here is that with the time, we are developing the drugs, right, and these drugs are actually evolving the resistance because of so many reasons, which I am not going to discuss anyway. So what you see here is that as the science is progressing and we are trying to develop the drug and trying to defeat the malaria parasite, the parasite is actually acquiring the resistance in the very short span of time. Okay, so if you see how much time it took for developing a resistance against the quinine, it took almost 200 years for a quinine, or more than 150 years for the malaria parasite to acquire the resistance against the quinine. But it took less than 20 years for it to evolve the resistance against the chloroquine. And you can see the data is given for even other drugs also. So the ability of malaria parasite to acquire the resistance against these drugs are very, 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 at an alarming stage, right? And that is because of the inappropriate usage of these drugs or many kind of, aspects. And because of this only, the WHO recommends that you should follow the ABCD of malaria parasite, where you actually aware or you basically spread the awareness about the disease, so that people should know these are the symptoms, these are the way you can protect yourself. Then you should also protect the, one of the very important thing is what people have done the clinical trials even in the Africa and other countries, that if you are even just staying in the mosquito nets, if you're just installing the mosquito net in your home, you are actually reducing the malaria cases very drastically. And then we also have the pneumophylosis and diagnostics, and all these steps are being very, very important as far as the protection from the malaria is concerned. Now talking about the drugs, right? You develop the drugs and then the drug is actually you injecting the drug to the patient, right? And what happens when you are injecting the drug into the patient is it is interacting not only with the target sites, it also goes to the off-target sites, such as I have taken an example of artemisinin. If you are injecting the high doses of artemisinin, it is going to affect the central nervous system, it's going to affect all sort of other organs. And so with the, and this is not a story for artemisinin, this is the story with other anti-malarials, even with the chloroquines and other molecules also, they are actually hepatotoxic, they are actually causing the problems in the other organs. So with the anti-malarial drugs, we are facing the two problems. One is drug resistance, the other is the toxicity. And when we started working on this particular aspect, we thought of can we deal both of these aspects, and at the same time when we thought of initiating some work. So I gave this topic to one of my PhD students, which is Ina Dolwani, right? And then we thought like if we can do something so that we can be able to utilize it, because if we understand how the drug resistance is being evolved, we have taken the artemisinin as a model molecule, then you can be able to do something so that the same drug can be reused, can be used without even taking care of the developing the new drugs. And if you take care of the toxicity issues, you can be able to overcome this as well. So what we found is that if you are exposing the liver cells to the artemisinin is causing the dose dependently, there is a reduction in the cellular viability, and it is also affecting the cellular integrity, and it is also causing the leakage of all the liver specific marker enzymes into the supernatant. What we also found is that the artemisinin halts the migration of mammalian cells. So remember that the mammalian cells have an integral function of moving from one part to another part so that they can be able to, they crawls and goes from one part to another part of the body, and that's how they actually convey the messages. They do a lot of, remodeling and all that. So artemisinin actually stops that kind of migration as well. So, and then what we did is we took the artemisinin and we blasted the artemisinin into the different types of drug target databases. And what we found is that it is recognizing the different types of proteins from the human database, okay, and majority of these data proteins were belonging to the different types of the pathways, right? Whether the pathway is related to the cell signaling pathways or DNA repair pathways or cell cycle pathways and so on, and they were distributed throughout the different components within the cell as well, right? So some targets were related to the nucleus, some targets were present in the mitochondria, and all other kinds of things. Not only that, these components or the proteins what we have identified were also interacting with each other, and that's how they were making an interactome. And this interaction is very, very important for the artemisinin to cause the artemisinin-induced toxicity. To test this, whether these findings are perfect, what we did is we modeled these proteins, or we actually extracted the structures from the database, and then we did the docking of the artemisinin into these. Okay. So what we found is majority of these proteins where the artemisinin was docking very nicely, and the artemisinin was making a lot of interactions. And these complexes are stable when we are done the molecular dynamics stimulations and molecular dynamic stimulations was saying that the artemisinin is fitting nicely into the active site, and then it is staying for a very, very long time into the active site. That means these are actually the targets for the artemisinin, or these could be potentially be the target for the artemisinin. To test these things, what we did is we also tested of these, so these proteins were belonging to the different types of pathways. For example, the cyclin B1, right? So cyclin B1 is a protein which is regulating the cell cycle, and some other protein as well. So what we ask is, if this is the case, it should actually affect the cell cycle of the affected cells. And we found that if you are exposing the cells with the artemisinin, it is actually halting the cell in the S phase and does not allow them to grow, right? And the same is true for the different types of proteins. So what we found is, if you expose the cells with the artemisinin, there is a upregulation of the different types of cyclins, there is a upregulation of the majority of the proteins which are associated with the apoptosis and so on. So that is a very preliminary study which we are, with which we can be able to say that artemisinin is utilizing the multiple targets, and that's how it is actually affecting the different types of molecules. Now, if we can be able to identify these molecules more in an exhaustive way, we could be able to, overcome the toxicity issues by developing the adjuvant molecules, so that you can be able to reverse these effects. And if we can be able to overcome with the toxicity of the artemisinin, we could be able to, utilize even a higher concentration of artemisinin. And if we can be able to utilize the higher concentration of the artemisinin, we somehow could be able to even overcome the other risks. Now we want to shift to the Plasmodium falciparum, or the malaria part, right? So one component is we would like to understand the toxicity. The other component is we also want to know how we can be able to improve the anti-malarial activity of artemisinin. And that is the only possibility, or there is only possibility you can actually be able to improve the drugs if you can be able to identify the targets. And you will be surprised to know that not even a single molecule of anti-malarial drug, what we use or what has been already been used in the past, is directed against any targets, right? So when I was working in CEDARI, right, and when I was working in Professor Dr. Puri's lab, right, we were getting a lot of molecules from the chemistry department, but they were not against a target. They would be like randomized molecules which we were screening, and then say, okay, this is the molecule better than chloroquine, and let's go to the. So that approach gives you a very good anti-malarial drug, but does not give you an opportunity to improve. So we thought, why not we should give a shot if we can be able to identify the targets of the artemisinin in the malaria parasite. And this is the approach what we have used, right? So we took the artemisinin structures. So artemisinin has the different pharmacophoric properties. So we have dissected the artemisinin into the different types of pharmacophoric zones. Then we took those molecules and blasted into the drug banks and the Swiss target predictions. Then we got some proteins, right? So these proteins were belonging to the humans, not for the parasites. And then we tried to identify the homologous proteins into the Plasmodium. Then we verified these studies using the help of the molecular darking and molecular marking studies. Then we found the final targets, and then these final targets were verified by the in vitro studies. So what we did is, what we found is this. Okay, so artemisinin is basically hitting these many number of proteins if you go with this particular approach. Okay, so the number was very huge. Okay, and what you see here is that the artemisinin is affecting the chunks of huge amount of targets. It is actually affecting the metabolic pathways, it is affecting the cell signaling, cell cycle, and so on. So all these proteins were actually belonging to these particular type of physiological pathways. Now looking at the which pharmacophoric molecule or pharmacophoric point is recognized by which target, so we classified them accordingly. And then based on this, we actually did several studies of making these molecules. We prepared a molecular model of these proteins to test whether our finding is correct or not, right? And then we did the simple molecular modeling studies, and we found that most of these molecules were binding the artemisinin, right? And the artemisinin was fitting nicely into their active site, making a lot of interactions and so on, right? But that does not prove or does not conclusively prove that these proteins could be the target of artemisinin. So then we focused on one of the pathways because the artemisinin is affecting the metabolic pathway, right? So then we focused on one of the pathways which is being called as hemoglobin degradation pathway, and we found in our searches that there are a lot of proteins which were belonging to this particular protein, this particular pathway. And then one of the proteins that we have extracted from this pathway is the plasmapsin-2. The plasmapsin-2 is one of the important protease which is responsible for hemoglobin degradation into the food vacuole, and then that actually provides the nutrition for the parasite to grow. So artemisinin was fitting nicely into the plasmapsin, and the binding was stable in the using the molecular dynamic studies. And simultaneously, we also cloned the plasmapsin. So plasmapsin is a monomeric protease which is present into the lacunar falciparum, and plasmapsin is active against the non-specific proteins or even the specific protein like the hemoglobin. And then the first question we ask is whether the plasmapsin is going to bind the artemisinin or not, because that is what so far what we were doing is we were doing the computational studies to say, okay, artemisinin is binding into active site or not, right? So then we did isothermal titration calorimetry, right? And what we found is that the artemisinin is binding into the active site of the plasmapsin, right? And most evidently it is showing a very nice, binding curves. Then we went on and say whether the binding is within the active site. So if the binding is within the active site, it should disrupt the plasmapsin's protease activity. So plasmapsin is okay. So plasmapsin is known to degrade the hemoglobin. So first we characterized our protein using the hemoglobin degradation assay and using the SDS page, right? And what we found is that plasmapsin is degrading the hemoglobin, and then we added the artemisinin into the reactions, and surprisingly, artemisinin was, inhibiting the plasmapsin-2 activity, and then we asked whether the artemisinin binding is reversible or irreversible. So what we did is we exposed the enzyme to a very high concentration, very small amount concentration in a small volume, so that effectively our artemisinin will be very high. And then we diluted the reaction to see whether the activity will regain or it will get inhibited. So it is a reversible inhibitor. As soon as we diluted, the enzyme has regained its activity. Then we did some computational studies to see whether the artemisinin is competing with the hemoglobin peptide or so. So plasmapsin is actually degrading the hemoglobin, and it is generating a lot of peptides. So we took one of the peptides, we did the molecular modeling and asked whether the artemisinin could be able to affect the binding of that particular peptide. And surprisingly, it is doing so. It was replacing that particular peptide and does not allow the peptide to go and bind into the active site. That is being verified with the help of the that is being verified with the help of the molecular dynamic stimulation to see whether the complex is stable and that particular finding is correct or not. And then eventually we did the kinetic studies of whether the artemisinin is inhibiting the plasmapsin-2. So artemisinin is inhibiting the plasmapsin-2 in a...
If you could wind up in next 3 to 4 minutes.
Yeah, sure. So with the so plasmapsin is artemisinin is inhibiting the plasmapsin-2 in a competitive mode, and both the Michael Momentum and live over plot is saying that, right? And so with this, we are hopeful that we could be able to reactivate the artemisinin. We could be able to take care of the toxicity as well as the resistance issues related to artemisinin, because that is very important. Developing a new molecule is a very challenging task, right? Compared to that, if you can reactivate and you can regain the activity of a particular molecule. And this actually gives the new hopes for the anti-malarial drugs. So we have taken the artemisinin as the model molecule. These studies could be replicated even with the other anti-malarial drugs or even the combination of the drugs, and that could actually be able to take care of the drug resistance and as well as the toxicity related issues. At the end, this is our malaria group. So we actually have multiple groups in the lab. So we have a group which is working with the malaria parasite, and there's another group which is working with the cancer also, and so on, right? So, and this is the Ina Dodwani, and this is my group, and this is our the Department of Biosciences and Bioengineering, and this is the beautiful campus of IIT Guwahati. Now, if you have any questions, I'll be happy to answer.
Thank you, Professor Trivedi, for that very interesting talk. We have time for a quick couple of questions. If there are any questions, please. Until we have any raised hands, if I may quickly ask Professor Trivedi, one of the concerns of these anti-malarial molecules, for example, we know for chloroquine also, the safety window is very low, right? For chloroquine also, it is the margin of safety is less than 10 folds. So, is there any way forward or anything that could be done on the chemistry side towards further modulation and increasing that safety margin for any of these anti-malarial molecules that you're looking at?
I think that probably the chemistry part I don't know. Yeah.
Okay, so any chemistry modification like scaffold, any particular toxicophores that could be removed, or any other pharmacophores that may have any? Yeah, Professor Tripathi, you want?
This is a wonderful one. Artemisinin is a very good drug for the patients suffering from some enzymatic diseases like that. So this is the best alternative drug for the patients suffering from D6PD and like that. So it's a wonderful. What is the what you want to optimize by using this study? How much target can be achieved by, because in all the quinine drug derivatives they are having the hemoglobin degradation and hemotoxicity. What exactly is from your studies can be concluded for us we can develop that?
See, initially our purpose is to understand the toxicity. If we can actually elaborate the same and see whether the artemisinin is hitting some of the targets which are crucial for the cancer, probably it can be exploited for that way also.
One more thing I would like to mention that artemisinin is 1,2,4-triazole ring is there. Yeah. Did you study the role of triazole ring in the toxicity? Did you study the role of triazole ring in the toxicity?
No, we use the whole molecule.
Responsible for antimalarial activity. That 1,2,4-triazole ring is responsible for antimalarial activity.
Okay.
And have you studied through your study that this triazole ring is responsible for any toxin?
But that kind of infrastructure we don't have. We cannot synthesize the parts of the molecule. We can only get.
Have you studied the toxicity study of 1,2,4-triazole ring in your study?
No, we don't.
In recent years also, this triazole ring, people are preparing dimers, trimers, several modifications in the artificial already. You are planning to study those things?
No.
That's what his forte. So he's a chemist by training. So yeah, that would probably. Yeah.
Chemistry part, people are doing computational.
Thank you, Professor Trivedi. That was a very interesting talk and it evoked quite a bit of discussion. So thank you so much. Round of applause for Professor Trivedi. Over to Professor Khanna to introduce the next speaker.
With this, we now move on to the next presentation, and I invite Dr. Prabodh Chand Sharma. Dr. Sharma is Associate Dean at Delhi Pharmaceutical Sciences and Research University, and he is a leading pharmaceutical scientist and has over 16 years of research experience, and has also been associated with Panacea Biotech. His contributions are focused on the design and synthesis of novel tetracyclic compounds with anti-infective and anti-cancer potential. He has published over 135 papers and also contributed many book chapters. He has supervised and mentored many PhD students and also Alpharma. And with this brief introduction, I request Dr. Sharma for the talk. Dr. Sharma, you can take 5 minutes.
Yes. 15 minutes? 10 minutes. Okay, thank you. Yeah. Honorable Chairman and Co-Chairman of the session, distinguished audience present here, and the organizers, thank you very much for providing me this opportunity to interact with this gathering here. And I will be talking about sustainable chemistry and its applications, and at the last briefly what we are doing in our laboratory. Am I audible?
Yeah.
Okay. So all we know that chemical and pharmaceutical industry is under high strain due to increasing demand of pharmaceutical and chemical substances, and there is an enormous strain on the pharmaceutical and medicinal chemists for providing new chemicals and new in bulk quantities as well. And with traditional resources, there are several gap areas like non-renewable sources which are available in a very finite quantity, which is likely to be depleted, and exploitation becomes non-sustainable, and the sources are becoming scarce, and in addition to that, huge quantity of waste is generated, which is allowed to accumulate in feeding landfill sites, and these scenes are common nowadays. I am from Delhi, and you can see such type of mountains of garbage, and the pollution that we are facing, it is becoming day by day a very big problem. So to address these needs, the principles of green chemistry were proposed, because there are primarily 3 types of hazards: health hazards, physical hazards, and environmental hazards. So Anastas and Warning, they proposed the principles of green chemistry to fill the gaps, and primarily by the use of green solvents, green catalysis, efficient processes, environmentally benign products, and green and renewable raw materials were employed for making the chemistry sustainable. There are 12 principles of green chemistry. It primarily focus on pollution and accident prevention, and on energy and resource sustainability, and on safety and sustainability aspects. So the current trend is moving from the traditional economy, which is linear economy, where we use a product, we take from the environment, make some product from there, use the product, and dispose the product after its use, and ultimately pollute the environment at every stage. So that is the concept for sustainable chemistry is right from the beginning. You make the product, use it, reuse the product, then remake something from the used product, and finally recycle it and again use for further some commercial purpose. So this is the concept of circular economy, and green chemistry is based on that to design, and it aims to design sustainability right from the beginning of the chemical process, and preventing the waste formation at each step. And it's always better to avoid the formation of waste rather than cleaning it at the end. And atom efficiency is very, very important. I will be taking few examples from pharmaceutical products, which needs to, the atom efficiency needs to be addressed, and energy requirements are also to be minimized. So we should, and definitely discouraging the use of volatile organic solvents has been undertaken in sustainable chemistry. So in the concept of sustainable chemistry, products are now increasingly treated as a resource equal to raw material, and developing processes where stoichiometric reagents are replaced with the catalytic reagents, and end products are becoming they are biodegradable, leading to less hazard to the environment, and analytical methods follow real time in process monitoring. So the role of a chemist is considered to be making a world of chemistry without generating any type of waste. And this concept of green and sustainable chemistry has been successful in several aspects, right from reduction in the use of volatile organic carbons up to 50% and reduction in other hazardous materials also. We'll be taking one case study where production of ibuprofen has been improved. So traditionally it was discovered in 1961 by Stewart Adams, and he was a pharmacologist working with Boots UK company, and this process of manufacturing of ibuprofen, it was having several limitations, like there were 6 chemical synthesis steps involved, and there was production of 9 different type of byproducts which were hazardous to environment, and the one major challenge was production of aluminium as a byproduct, and it was disposed in the environment. And it is considered that depletion of the forest in Central Europe was attributed to product so much byproduct aluminium which was produced in the formation of ibuprofen. And moreover, it was having health hazards also, and there are several type of toxicities were associated with aluminium. So the BHC group has undertaken the alternative sustainable green method for synthesis of ibuprofen, where the chemical reaction was reduced to 3 steps instead of 6 steps. So that was their achievement, and there was technically no byproduct formed, only acetic acid, which was further used for some other chemical reactions. And the atom economy, which was less than 40%, was increased to 77% in this case. So this is one of the most successful examples of sustainable chemistry in pharmaceuticals. Another example of successful example of application of sustainable chemistry is related to synthesis of letermovir, which is an antiviral drug. And this was a very, very important drug, and it was given orphan product designation by US FDA. But the problems associated with this drug was that the yield was only 10% with the traditional method, and there were 9 different solvents involved in the synthesis of this drug, and high palladium loading was done, and it was required in Hack coupling. So alternative methods were designed for increased atom economy, and for this some deviations were done, and a new method was developed where Aja-Michael reaction was carried out for synthesis of letermovir, and overall increase in yield was from 10% to 60%, and there was a reduction in raw material cost was up to 93%, and water usage was reduced up to 90%. And it is estimated that over the life cycle of this drug, there will be reduction in 15,000 metric ton of waste, and carbon footprint will be reduced by 89%. And for this study, for this sustainable chemistry discovery, Greener Synthetic Pathway Award was given to BHC company. Now, the role of biocatalysts is very important in synthetic processes, and we know enzymes are the biocatalysts, which are nature's catalysts, which are not only biodegradable but renewable as well, and they have ease of production and they can be produced by fermentation, etc. So by application of biocatalysts, pregabalin was synthesized, and with pregabalin, with the modern method of synthesis of pregabalin, 90% reduction in solvent usage was done, and there was 50% reduction in the requirement of raw material. And the reduction was such that it is equivalent to removing 1 million cars from the Indian roads. So that much reduction in pollution was obtained with the sustainable chemistry approach for synthesis of pregabalin. And the next aspect is related to plastics, which are having a bad reputation due to non-renewable petrochemical products, and they are not biodegradable as well. So, as an alternative approach to synthesis of plastics, bioplastics have been developed, and there is a startup company with named New Light Technologies, which did not change the process, rather they changed the use of biocatalyst. They used a proprietary biocatalyst for synthesis of bioplastics, and this increased the yield to 9 times than the original. And the cost was reduced by a factor of 3, and thereby making air carbon, which is a bioplastic, which is cheaper than the oil-based plastics. And interestingly, this air carbon is used by many of the world's leading companies, not only pharmaceutical but many other companies also. And due to this alternative biocatalyst approach, the award of most innovative company was given to this company. And this market of bioplastics is increasing day by day, and it is expected that it will capture up to 40% of the plastic market by the next 5-6 years. And then use of alternative solvents is also very, very important and getting popularized in sustainable chemistry, because the solvents are very important in chemical synthesis. They are used to dissolve solids, reduce viscosity, modulate temperature, and to recover the product. They also affect the chemoreduced selectivity of the reactions. But unfortunately, the traditional organic solvents are associated with so many ill effects, and use of alternative solvents in sustainable chemistry is gaining popularized. This is known as solvent substitution. I'll cite very interesting example of fabric industry, where drying of one t-shirt takes around 7 gallons of water. So first is use of high quantity of water, and second is the energy required for drying the t-shirt or the cloth. So alternative approach to this was taken up by a Dutch company, and they initiated water-free dyeing by using supercritical carbon dioxide as a solvent instead of water. And due to that, there was no drying required, so water wastage was removed, as well as no energy was required, so energy has also been saved. So there are some other examples also, like bio-friendly paint removers, firefighting foams, and some other examples. And to enhance the sustainability in the chemistry, now artificial intelligence-based methods are being employed. In our institution also at Delhi, we have initiated a course with the name Artificial Intelligence in Medicine Master's Program, and that group is working well for design of drugs based on artificial intelligence. But if we talk about material science and sustainable chemistry, so IBM Research Foundation have used AI to find a new family of recyclable and self-healing polymers, and AI algorithms have greatly speeded up the process of finding new materials while guaranteeing the environmental compliance. Similarly, BASF, the same company which was, I have cited an example for sustainability and production of new sustainable molecules. So they have developed AI-driven procedures to improve its operational efficiency. And by using machine learning algorithm, BASF is able to forecast and modify energy consumptions instantly, resulting in significant decrease in both carbon emission and energy expenses. So the process of adoption of new methods, sustainable methods in chemistry with green processes using AI is very now taking too much popularity, and Merck and Pfizer are the leading groups which expedite the medication development or drug development process and eliminate the need of hazardous solvents and energy intensive reactions. So there are certain barriers also associated with the introduction and use of the sustainable chemistry in the industry, because the regulations are there which are being followed, and if they adopt the sustainable practices, the entire process will have to be recertified. And the changes are benign processes, and they are inhibited by cost-intensive control-oriented regulations. Moreover, with the business entrepreneurs, there is a lack of awareness, and actually we need the knowledge of engineering, biotechnology, economics, AI, and above all toxicology to integrate the sustainable chemistry for industrial applications. And the methods have to be economically lucrative also, and there is a capital investment to introduce. So these are some of the barriers or hurdles, but still if there are there is sound scientific knowledge, so we can have, time has been shown, so briefly I will show our efforts. So this is our group working on sustainable chemistry. Yeah, I'll take two minutes more. Yeah. So this is our group working on sustainable chemistry at Delhi Pharmaceutical Sciences, and we have been able to substitute the chloramine T as a catalyst in the reaction for facile one-pot synthesis of certain heterocyclic compounds, and the results were very, very interesting. Furthermore, synthesis of anti-infective fluoroquinolone derivatives for antibacterial activity employing green chemistry approach has been carried out. Effectively, microwave induced synthesis has been done in our laboratory, where the reaction time has been reduced from 3 to 4 hours. It has been reduced just to 60 to 90 seconds, thereby reducing the reaction time, reduction energy consumption, and increasing the yield quality as well. So these are some other examples of sustainable chemistry work carried out at our laboratories. And this is one recent work where we have been able to develop ionic liquid carbon rods derived from Tacoma strains, and this was specially developed for multi-drug resistant strains. So with that, thank you, and if there are any questions.
Thank you, Dr. Sharma, for a wonderful talk. You can take one or two questions, quick questions. And if not, where do we stand as a country in using the green technology and its application?
It's a very right question. We, as I said, the regulatory constraints are there for industrial application of these technologies. Although if we talk about academic and research endeavors, we are doing reasonably fine. I will not say best, but we are doing reasonably fine. There are so many new endeavors. But when it comes to the industry to adopt the process, they have to have entirely new certification. It is cost intensive as well as one-time capital investment is there, and the commercial aspect, that gap is there, and yes, we still have to do a lot in this.
In the interest of time, I will not go I like to discuss after the session also, but this is one of a very good idea this particular shared with us. Thank you very much.
Thank you.
Thank you. Moving on, we have an oral presentation now,
Yes, sir.
right?
Yes, sir.
Okay, so may we now invite Dr. Ratna Pandey to deliver her talk. She will be talking about the rhetoric of AI in medicine, hype, hope, and reality.
Good evening, everyone. I'm Dr. Ratna Pandey, and I'm from Dr. Bhimrao Ambedkar University, Agra. So since morning I'm here. At 5 o'clock I started. I'm just waiting for my turn. But it's a privilege being a part of this conference, and it's an honor to hear such great speakers. And I would seriously thank the Department of Pharmacy for organizing such a wonderful conference. And of course, I would like to thank Taneja sir is not here. He is also, earlier he was in the Agra University. So a very nice gentleman, and it's a privilege that he invited me. Before starting with this, my topic is the rhetoric of AI in medicine, hype, hope, and reality. Before starting with my topic, I would just like to draw the attention of all the students sitting at the back. Just can you ask that fellow to please, yes, please raise your hand, right? I would like all of you to just go through this slide. It is just a matter of 10 or 15 slides I have, and very quickly I'll finish. The thing is, I would like to know from all of you, are you all aware of AI? Yes? Yes? Are we all diving into AI? Yes? And what I felt is that in medicine also AI is playing a lot of role, right? So I would just like rhetoric. Is anybody having an idea of the term rhetoric? Rhetoric, I'll tell you. मैं आपको हिंदी में समझाती हूँ. जब हम शब्दों का चयन करते हैं, when we use words, right? And we use words and also orally as well as in written form. But we are using words, but we are not actually. We are using words just to impress, right? So this rhetoric is to impress people, right? And the sincerity is not taken in concern. So sincerity कि हमने क्या बोला, क्या लिखा, sincerity नहीं है, but impressive बहुत है, right? So what happens that we all get attracted, right? So let's jump into it. The topic है कि AI कैसे, किस तरह से rhetoric of AI is taking in medicine, taking part in medicine. It is creating hype. Hype, you all understand? Yes, बहुत ज़्यादा बड़ा चढ़ा के दिखा रहा है. And then there is hope. A lot of hope has been shown to us, and we'll talk about reality, right? So quickly, I'll jump into it. So the agenda is, of course, I'll be giving. I've already introduced the questions which I have taken. Rhetoric of AI in medicine. Hype. Hype is the language of promise and speed. कितना बड़ा चढ़ा के कि स्पीड कितनी अच्छी होने वाली है इस मेडिसिन में, और प्रॉमिस है कि कुछ ना कुछ अच्छा होने वाला है. देन द लैंग्वेज ऑफ होप, द लैंग्वेज ऑफ कोलैबोरेशन एंड प्रोग्रेस. सो प्रोग्रेस भी होगी एंड देयर विल बी अ लॉट ऑफ कोलैबोरेशन. एंड व्हाट इज द रियलिटी? The language of caution and responsibility. So we'll have to take care of, we have to be cautious as well as we will have to be responsible enough, right? So there is a comparative finding as well, and then the conclusion. So now, AI is transforming how drugs are discovered, developed and delivered. Beyond technology, the language around AI shapes how we perceive, progress and also take care of ethics. The study explores how different stakeholders, there are a lot of stakeholders in the world of pharmacy. कौन-कौन है stakeholders? First is media, the second is the corporate world, then there are scientists, and then there are the policy makers. So how they use language to frame AI's role in medicine. Now, to analyze how rhetoric constructs public understanding, किस तरह से लोगों पे impact पड़ रहा है, किस तरह से how people are understanding the concept and how they are building in trust. So these are the research questions which I have worked. How do linguistic choices in media, scientific and institutional discourse influence public understanding of AI in medicine? And then what rhetoric patterns define the hype, hope, reality, and it is continued. How can language finally be used ethically? Now, ethic is very important to bridge the gap between innovation and understanding. Now, so these are some methodologies which I went through. So I took the approach that is qualitative, rhetorical, and discourse analysis. I went through now the data size which I would like to discuss is I went through some 19 texts. And through this, I have taken out my research work. And ultimately, the goal is that how linguistic tones from journalistic hype to academic realism, and understand how rhetoric shapes trust and perception of AI in medicine. So these are some of the theories I went through, some of the coding and some of the purpose. Now the purpose is to interpret how linguistic patterns shape ethical and emotional perceptions of AI's role in medicine. Still, I was just talking to one of the teachers sitting here, and I asked, Madam, क्या अभी पढ़ाई फार्मेसी की हिंदी में शुरू हो गई है? And still she said no. अभी भी फार्मेसी की पढ़ाई में अभी हम हिंदी में नहीं आए. Although in some places engineering work has been started, that हम हिंदी में पढ़ाना शुरू कर दिया है, but still फार्मेसी में अभी भी इंग्लिश का रोल बहुत important है. Right, and of course I'm a faculty of English, although I was not a part of this pharmacy world, but I thought of jumping into this and I really liked it. So what is hype and what is the rhetoric of hype? Now, language of speed and transformation. So now we are using it, the language and how we speed up. So this is how the hype is created कि किस तरह से कि कैसे AI काम कर रहा है, कैसे मेडिसिन वर्ल्ड में कितने ट्रांसफॉर्मेशंस हो रहे हैं, कितने रिफॉर्म्स हो रहे हैं, राइट. सो हाउ रिवोल्यूशन इज बीइंग क्रिएटेड, क्या अनकवर हो रहा है, क्या डिस्कवर किया जा रहा है, एंड हाउ इट इज डिजाइंड. सो एवरीथिंग थ्रू द हेल्प ऑफ AI इट इज बीइंग शोन. सो AI इज पोर्ट्रेइंग एंड इट इज लाइक इस तरह से डिपिक्ट किया जाता है कि AI इंडिपेंडेंटली एक प्रॉब्लम सॉल्वर है, बट इन रियलिटी इज इट सो? क्या ह्यूमंस की जरूरत नहीं है? Yes, we all need pharmacists. Yes, young minds are sitting here. All are required. So, example, BBC News ने कहा कि AI designs antibiotics for superbugs. So, this was the recent news. And then in the story works, it was said speeds up the process of discovery. So, what in reality? Oh, okay. In fact, it creates excitement but simplifies complexity. And what is hope? What is rhetoric of hope? Language of collaboration progress. AI is AI as assistant. It is working as partner, and it is not the replacement. Right? So some people are saying that it is in the air that the jobs of pharmacists and the engineers will be taken up by AI. Yes, people are nodding your heads. Yes, but it is not so. It is just that we have to upgrade ourselves. So some of the experiments I have been told I have to hurry up. Okay, it of course hope एक create दिखाई दे रहा, hope है, the hope is there. It builds trust and encourages responsible optimism. But coming to reality, what actually is reality? Academic tone, we have to be cautious, we have to be precise, we have to be grounded, and we have to focus on reproducibility. कि क्या हम produce कर रहे हैं, उस पर reality check करना बहुत जरूरी है. The transparency has to be there, and human insight has to be there. Yes. So, for examples, nature medicine, no clear definition of AI discovered drug. So this is the reality, but हम hype कर करके show करते हैं market में. Yes, some of the other research work said walls of magical theory of AI in medicine. So there is again a warning. And then AI should assist rather than replace clinicals. So again, you don't have to worry, right? So you people are safe enough. So of course, the effect is that restores ethical and scientific balance. This is the reality. So this we have talked. So this is the comparative study. Now what is the finding? Media, of course, what does media do and cooperates? What they do just to उनका एक वो बन गया है कि market बढ़ाना है अपना. So they use too much of hype, right? So what they do, they use too much of such terms through AI. They generate terms so that the market is raised. उनका PR बहुत ज़्यादा बढ़ जाए, commercialization is increased. And then academic and policy makers, of course, they neutralize, they are the voices, act as corrective narratives. They give the corrective narratives, and then language directly shapes trust, funding priorities, and ethical perception. Ethics has to be there, ethics cannot be forgotten. And ultimately, the hype, hope, and reality framework reflects how AI transitions from innovation to institution. So these are the implications we must take care of. For science communication, journalists avoid exaggeration, favor clarity. Exaggeration जो है बहुत ज़्यादा बढ़ा चढ़ा के बोलना, that has to be avoided by the media and the journalists, right? And the corporates, they will have to balance the promotion with transparency. That transparency at times is missing. Then the researchers, what they have to do is they have to communicate uncertainty ethically. So ethics has to, again, as I said earlier also, ethics has to remain. We cannot just finish this ethics. We have to take care of this ethics. AI, okay, fine. And what is the core message? The progress is not only technical, it is linguistic. So not only technical, with the usage of words also, the pharmacists are doing great work. So the role of English has to, the role of language has to be taken care of. That is all. Thank you so much. Oh, I'm so sorry. Fine, thank you all of you.
We can take a quick question if there is any.
Yeah, at the back. Please, yeah.
Ma'am, as you told that AI is a helping tool, but on the other hand, it's a proper drawback of AI.
This is what I'm saying, that we have to be very careful while taking the help of AI. This is what ultimately I'm trying to say, that all the students especially, you don't get too much impressed by AI, that AI, whatever AI has said is correct. Right? So this is what I'm saying, that we as students have to be very careful while using AI. Don't get आप इसमें चक्कर में फंस मत जाइए कि AI ने क्या कह दिया, सब सही कह दिया. This is what I'm trying to say.
This AI also needs a human.
Yes, of course. This is what I'm saying. You people are safe. आपको अपने को वो groom करना है, groom up करना है. You are not at all going to get affected. But हाँ, आप बिल्कुल totally given हो जाओ कि हमें AI के थ्रू ही सारे काम करने हैं. AI जो कह रहा है वो सच है नहीं. AI को चलाने के लिए, use करने के लिए भी एक human mind चाहिए. Right? So, chill.
Okay, thank you.
Thank you so much.
Thank you, Dr. Ratna, for a nice talk. I think there will be a lot of debate. And people will take advantage of our presence and will not leave even the dinner also. With this, I'd like to inform you that the organizers have been very kind to accommodate one more presentation here, and for that, I have pleasure to invite Dr. Devdutt Chaturvedi. And Dr. Devdutt is working as professor of chemistry and head Department of Chemistry at Mahatma Gandhi Central University, Motihari. And he has essentially contributed in the area and is very well decorated, has essentially published and recipient of many awards and has published extensively in peer-reviewed journals. In the interest of time, I will not go into the detail, but will request Dr. Chaturvedi just to give the talk. Dr. Chaturvedi, you have 10 minutes now from now.
Sir, 5 more minutes. 15 minutes.
10 minutes.
So, I'm very much thankful to Professor Taneja Saab, Vice Chancellor here. Very close associated with me, and also I'm thankful to Professor Salvi Tripathi, Professor Pushpendra Tripathi, and all the organizing team of the committee. And I would like to congratulate also very good conference over here. Actually, it's the first conference in this department. And I also I'm very much thankful to Professor Khanna Saab and my friend Dr. Ahmed Najib, who Khanna Saab actually introduced me well. So it's very interesting topic today. It's just I have presented the different things in a single way. You see here I have taken the topic that is heteroaliens. Heteroaliens is just the chemistry of heteroaliens when the replacement of allene. This is allene, here. This allene. Yeah. When the replacement of this carbon, allene carbon with the heteroatom, then it leads to the heteroaliens. Very simple molecules like carbon dioxide, carbon disulfide, carbon sulfide here. So, in overall, we will be discussing some of the chemistry of these different kind of heteroaliens. You see here, these are the alternatives for the synthesis of various molecules. You can see what say alternatives. And you see, we have developed several methods. One is the soap agent, carbon dioxide, carbon disulfide system. Another is trichromacy, CO2, CO2, and COS system, using olefin and CO2 and CO2 system, and copper polyene and composite. The initial strategy we have taken here, just by reacting 2 moles of amine with carbon dioxide, there is the formation of monoalkyl carbonate ion, and in presence of this carbonate ion, this O- will attack on the alkylic agent of this carbon, leading to the formation of carbonate. The formation of carbonate may be enhanced by the reflectivity of this ion, and there are many phase transfer catalysts and the basic catalysts we have used here to generate the carbonate. Because the target of my process is about cow milk. So next, so we have developed several methods over here using various kinds of alkaline agents like alkyl or glucosides. And adopting similar approach, we have used this amine carbon dioxide, and then this kind of monoalkyl, the monoalkyl diethyl ion by hydrogen. And in case of S minus will attack on this electron carbon, this leads to the formation of diethylcarbonate. And the reactivity of carbon disulfide is moved in the carbon dioxide. So all the reactions of the carbon disulfide were at room temperature. But in case of carbon dioxide, there are much subsisting to 90 degrees centigrade. Temperature we have used here. And here we have another reagent that is triphenyl phosphine dicarboxylate. This is known as Mishnopol reagent. And using Mishnopol reagent, we have generated sequence of different kinds of molecules you see here. So, already we told about the carbon. Actually, the bigger part we have started with carbonate. Carbonate are the stable class of the compound that from the listed carbonation by the substitution of amino in the study. And when this linkage is made in the cyclic system, there is known as the cyclic carbonate. And why we have synthesized these compounds? Because they have many potential applications here. You see here, they have many, like in synthetic intermediates, the pharmaceuticals, and the drugs and products, and in various kind of molecules. So carbonates play an important role in medicinal chemistry. And it was also realized that by incorporating carbonate into the molecule may lead to increase the manifold biological activity. Here, you see here, like the Texol, the carbonate is here. Many molecules you see here, carbonate increases the biological activity of the molecule. Several molecules here, we have already in the chapter, the more about many carbonates in the considered design, where we have realized that by incorporating carbonate may lead to manifold biological activity. There are many molecules you see here, heavy carbonates. So these for the different kind of activities, like you see here, this integral selectivity, this carbonate, many molecules over here. You see here potential integration carbonate based products. You see here the potential anti-terpene carbonates over here. Many kinds of molecules. You see here carbonates are synthetic additional molecules. People have synthesized carbonates in large base kind of protocols because carbonate linkage is very important linkage in increasing biological activity of the molecule. That receptors and increasing biological activity of the molecule. Like carbonate, we have synthesized diethylcarbonates. Like diethylcarbonates, we have here many applications in drugs, products like carbonates. Diethylcarbonates also in carbonates. There is alcohol by the oxygen. See here, diethylcarbonates also have many drugs in diethylcarbonates over here. You see here many drugs. So there are many drugs you see here. Here some diethylcarbonates over here also. So we have synthesized many diethylcarbonates. You see here we have used synthesized various kinds of diethylcarbonates. You see here we have synthesized carbonate from the starting point of alcohols and amines using carbon dioxide system. This is the material, but then this is the use of diethylcarbonates here. From the alcohols and amines. Synthesis of genthers using slow agent, using various kinds of private scale alcohols. We have synthesized the synthesis of diethylcarbonate agent. So we have synthesized also ASL type carbonates using slow agent. So we have also synthesized diethylcarbonates using slow agent using various kinds of ionic acid, carbonates and acid thiols. So diethylcarbonates using slow agent. So many compounds we have synthesized using different kind of chemistry. Like diethylcarbonates, carbonates also. Carbonates are the carbonates of hydrogens. So many heterocyclic compounds are being synthesized using catalytic system adapting similar approach which we have talked about earlier. So this is all about my work. This is very brief. Thank you.
Thank you, Dr. Chaturvedi. I think we are well on time. A big round of applause for Dr. Chaturvedi. And a very, very good. Not request answer. And if not, we'd like to thank you, sir, for being part.
Now I request our chairpersons to come on dais to felicitate our speakers. I would like to invite Professor Rajiv Jain. I would like to invite Professor Vishal Trivedi. I would like to invite Dr. Prabodh Chandra Sharma. I would like to invite Dr. Ratna Pandey. I would like to invite Dr. Ratna Pandey. Now I would like to invite Dr. Devdutt Chaturvedi. I would like to invite Professor Christine Kumar Tripathi, Director, Institute of Pharmaceutical Sciences, Lucknow University, to felicitate our chairpersons, Dr. Vinay Khanna, Dr. Amir Nazir, Dr. Vinay Khanna. Thank you, sir. We cordially invite all our guests for high tea at Guest House, and participants and delegates are requested to kindly proceed to Student Facilities Center for their high tea. Thank you.
Rajkamal.
Wait, 2 minutes. Wait.
As we are concluding day 1, now I request all of you to kindly give a standing ovation to National Anthem.